Hello, everyone, and welcome to NOAA's Omics Webinar Series. My name is Tracy Gill, and along with Catherine Egan, we are the co-hosts for this series. Before we get started, here are a few logistics, and most of them are in the chat box. Attendees are muted, but you can enter your comments and questions into the chat box at any time. The presenter today will address all questions at the end. If you want to go to a full screen or larger slide view, look above the upper right corner of the slides and find the button that has a box with a bracket around it. That box button toggles the full screen view on and off. There are two ways to access live closed captioning, and they are described in the chat box. If you are interested in getting a PDF copy of the slides, a link to the recording, and possibly a summary of the chat, we will send it out within a few days after the talk to all who are registered. If you are not on the weekly NOAA Science Seminar email list, but you'd like to be, please contact me at tracy.gill@noaa.gov, and I will add you to the list. And many thanks today to our captioner for making this recording more accessible. And now I will pass the mic to Catherine Egan, the Omics Coordinator in the NOAA OAR Ocean Portfolio, with the OAR.
All right, thank you so much, Tracy, and hello, everybody. Thank you all so much for joining us today. Welcome to the NOAA Omics Seminar Series. As Tracy stated, I am your co-host, Catherine Egan, the OAR Omics Coordinator, but I also sit in NOAA Ocean Exploration. The Omics Seminar Series is hosted by two groups, NOAA Ocean Exploration and the National Ocean Service through the One NOAA Science Seminar Series. Tracy Gill and I will be co-hosting the Omics seminars, and I will also be moderating the questions today. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and also highlight the incredible omics-related research that is currently underway both within and outside of NOAA. I'm also happy to announce that our seminars from this series are made available on YouTube and posted up on our Omics website a few weeks after the initial presentation. I will drop a link to that in the chat now, so you can view past seminars and also register for our next seminar, which I will discuss at the end of today's seminar. So with that, our speaker today is Dr. Nastassia Patin, and her presentation is titled Connecting Microbes, Phytoplankton, and Oceanography in the California Current with eDNA and Metagenomes. Nastassia Patin got her PhD at Scripps Institution of Oceanography, where she studied the chemical ecology of marine sediment bacteria and their bioactive compounds. She then did a postdoc at Georgia Tech in Atlanta, where she worked in a variety of marine systems to understand the role of microbiomes in ecosystem health and function. She focused on applying metagenomic and bioinformatic tools to recover high-quality genomes of uncultured marine microbes and link them to biotic and abiotic processes. In 2020, she joined AOML, where she currently works with Dr. Kelly Goodwin to leverage eDNA sequence data from the California Current to understand linkages among microbes, plankton, and metazoans, while advancing the omics efforts more broadly at NOAA. In her free time, Nastassia enjoys surfing, biking, cooking, and hanging out with her two cats. So thank you so much, Nastassia, for presenting today. I will now pass it off to you.
Thank you so much, Catherine and Tracy, for giving me this opportunity to present. I'm really excited to share some of the work I've been doing for the last year on eDNA in the California Current. I am part of the Atlantic Oceanographic and Meteorological Laboratory based in Miami, but I am physically stationed at the Southwest Fisheries Science Center in La Jolla. And today I'm going to be telling you about the California Current ecosystem, which includes microbes, phytoplankton, higher organisms, and some oceanography as well. So I want to get started with some acknowledgements. So I first want to thank my advisor at NOAA, Kelly Goodwin, who has really spearheaded a lot of these efforts and been a great guidance, as well as my colleagues at AOML, particularly Luke Thompson and Jean Lim. Our partners at the Monterey Bay Aquarium Research Institute, particularly Francisco Chavez and his lab members Nate Truelove and Katie Pitts. The team on the rockfish recruitment and ecosystem assessment survey cruise, which I'll talk about a little bit later on, where I got to do some field work this past spring. And finally, Aaron Satterthwaite and Chase James, who have provided some helpful comments and ideas as this work has progressed. So the overall questions and goals for this study really involve drawing connections between microbiomes and everything else going on in the oceans. So we know that microbial communities are really important for things like nutrient cycling and regulating interactions among higher organisms, but we don't really understand the specific links that drive ecosystem processes. So we're wondering how these microbial communities are first linked to just oceanographic processes like upwelling in the California currents. What are the relationships between microbial, plankton, and metazoan communities? And then finally, in a more bigger picture sense, which I won't specifically address today, but I would like you to kind of keep this idea in the back of your mind. How can we use water column microbiomes to inform predictive oceanographic models, whether that's related to climate or fisheries or other important applications? And we're hoping that these efforts will get us closer to those goals. So for those of you maybe not used to thinking about microbes as much, I just wanted to give a brief plug for them and why they're so important. So they are at the core, they're the base of the food web. So they lead up to the larger single-celled organisms, which then become the prey field for things for plankton, both zooplankton and phytoplankton in the ocean. They also are involved in interactions with plankton and fish, both in negative and positive ways. They can be pathogens, they can be crucial symbionts for higher organisms. But beyond those sort of basics, they are intimately involved in transforming the chemistry of the ocean, particularly their roles in nutrient cycling, like nitrogen, carbon, and sulfur are super important. And I'm going to highlight two examples that I'll come back to as sort of case studies for this importance. The first is the conversion of a sulfur-based compound produced by a lot of phytoplankton called DMSP. So microbes convert this compound to something called DMS, which is a volatile compound and has been implicated in seeding aerosol clouds and driving some climate processes. They also are involved in primary productivity as well as rhodopsin-driven autotrophy. So essentially converting energy using light rather than organic carbon. And they are also responsible for synthesizing vitamins and other micronutrients that may be in very low levels, but can really play a big role in determining health of things like fisheries and phytoplankton communities. So how do we study microbiomes? We use something called metabarcoding, which many of you have probably heard about, but just in case, I want to highlight that this is a process, a method involving marker genes where we sequence individual marker genes at a community level. And different marker genes are used for different types of organisms. So the 16S ribosomal RNA marker gene is the most commonly used gene for microbial communities. And then the 18S and 12S ribosomal RNA genes are used for phytoplanktons, invertebrates, and metazoans, respectively. And the CO1 cytochromoxidase 1 gene is used as well for different groups or different level of taxonomic resolution for phytoplankton and invertebrates. And then in addition to metabarcoding, I'll be talking about another type of sequencing called shotgun metagenomics, where we sequence the entire eDNA pool rather than just marker genes. And I'll go into the basics of these two methods and how they differ and why the different advantages offered by each one. So just to start with metabarcoding, for those of you not so familiar with it, the basic principle is that we have an environmental DNA sample, which consists of long pieces of DNA. They could be circular chromosomes, they might be linear. I'm just depicting them here as circular chromosomes. But from all of those different genomes, you can PCR amplify the specific marker gene you're interested in, get that all of those marker genes sequenced, and then that gives you an overview of the community in a survey, basically in a survey form. So you can see how much of your community is made up of your different taxa based on the differences in sequences. And once you have those data, you can start to look at differences between samples. So in this example, sample 1 and 2 are very similar to each other because they have very similar proportions of taxa, but 2 and 3 are quite different from each other because the same taxa are showing up at very different levels. So this is really just a way to get an overview of who's there in your community. And the main limitations to this is you only get relative abundances due to the PCR step. You don't have a sense of actual cell counts or biomass. And of course, as with any molecular method, there are some amplification biases. Despite that, it's been a really, really useful method for community overviews. So with that in mind, the first part of my talk will be linking microbial communities to water masses, so touching on the oceanographic component of this study. And I'll be talking about a set of amplicons from a 2018 field survey. So this particular field survey was part of the Monterey Bay Aquarium Research Institute's Cannon cruises, which they run a few times a year doing different experiments. And in this case, they did a cruise in the spring and a cruise in the fall. And the two cruise courses were different in kind of an important way. So in the spring, they ran a time course at essentially a single location. So you can see on the map here on the right, oops, where's my pointer? Yeah, on the map here on the right, the group of blue dots here were the cruise samples taken in the spring. You can see they're all clustered around a single location. And then in the fall, they did sampling along a front or along a water mass moving over space and time. And so although they covered a larger geographic area, they were essentially sampling from the same water mass. And so with those two experimental designs in mind, I'm showing you here an ordination plot of each of the samples that came from these experiments. And so in this type of plot, the axes are arbitrary, so don't worry about any specific variables that they represent. But what's important is the points that are closer to each other are more similar to each other in terms of community composition, and then points farther apart are more dissimilar. And so these points on this plot are colored by the depth from which they were taken. So we had a series of samples taken around 200 meters. And then a series of, most of the samples came from this shallow water column between 0 and 40 meters. And then the circular points are from the spring and the squares are from the fall. And so you can see right away that the big separations are by depth. So we have our deeper samples that cluster much separately from the shallow samples. And then also by season. So we have our square fall samples are really forming their own group over here compared to the circular shallow samples. So this will be a recurring theme in a lot of the slides I show, that season and depth are really driving our microbial community composition. So if we start to take a look at the top 5 taxa in each sample group, so don't worry so much about the names of these taxa over on the right-hand side. I mostly want to point out that in this plot, we have our fall shallow set of samples, where we can see that the groupings for these samples are pretty tight. So they're all fairly similar to each other across the samples, all the samples that were taken. Same thing for the spring and deep samples. But then when we get to our spring shallow samples, we actually see this wide variability in relative abundance of the top 5 taxa. And this makes sense when we think about the experimental design, where in the fall, they were tracking this single water mass over space and time, but really essentially resampling the same water mass over and over again. You can see that they're essentially getting replicates of the same water column samples. That's reflected in these tight groupings of relative abundances. The same is actually true for the spring deep samples, but this is the time course where they were in one location, over one time, but you can see that it seems like that deeper water mass was quite stable. They were essentially resampling the same water mass. Whereas in the spring, even though they were in the same location, they were sampling as different water masses moved through the surface ocean. And so you see a much more variability in the relative abundances. And so just to give you a sense of the kinds of things that we look for as microbial ecologists, who are these taxa and why might we care about them? So as an example, this genus called Amylibacter is part of a very abundant, widely distributed marine clade, commonly known as Rosiobacters. This particular genus is associated with diatom blooms and has been shown to degrade phytoplankton-derived polysaccharides. And as I mentioned in the introduction, this is a taxon that can convert the sulfur compound DMSP to DMS. So these are the types of functions and roles that these bacteria are playing, and why we might be interested in them oceanographically. In this case, this particular group, the marine group 2 Thermoplasmatoda, are an extremely abundant planktonic archaeal group. So now we're looking at members of the archaea rather than bacteria with no cultivated members. And this is another recurring theme where a lot of these taxa have not been cultured in the lab. And so the only way we can study them is through these culture-independent DNA-based methods. We have some taxa belonging to the Nitrosopumilus family, including the Nitrosopumilus genus. These are well characterized to some extent. A couple representatives are known to oxidize ammonia to nitrate, which is the first step of nitrification. But as we learn more about this group, we actually see that there are many representatives that are very poorly understood and probably are a lot more metabolically diverse than we currently know. So it's really crucial that we can study them, again, these culture independent ways. And finally, SAR11, this is the most abundant microorganism and other organism on the planet. These are extremely tiny cells with highly streamlined genomes. And they are woefully understudied due to difficulty in cultivation, as well as extremely high microdiversity within this clade. And so there's actually very few even metagenome-assembled genomes from SAR11 due to the computational difficulties. And I'll touch on that a little bit later on in more detail. Okay, so to go back to our amplicons, I want to point out that there's this one sample that seems to be a bit of an outlier. So this is a spring shallow sample that is clustering a little more closely with the deep samples than we might expect, especially from the spring subset. So I was interested in why this might be, and I took a look at the bathymetry of these two experiments from 2018. And again, the orange points show where the cruise was following a front, tracking a water parcel over time and space. And then the blue points over here are where the single point was being resampled over and over. And this is the location of this outlier sample. And I didn't see anything particularly interesting or different about this in terms of like being at the edge of a canyon, which might have a lot of upwelling, anything like that. But luckily, we have some chemical oceanographic data from the CTD casts that were done at this point. And so I took a look at those data. And so what I'm showing here are a few different plots. They all have just various chemical variables plotted on the y-axis. And so, for example, we have our nitrate, and I separated the points here into shallow and deep groupings. And what we can consistently see is that this outlier sample, although it is part of the shallow depth grouping, it is consistently at the lower, or in some cases upper end of the chemical measurements, showing that it's closer, in fact, in chemical composition to a deeper sample. So for example, it has the lowest oxygen levels of all of the shallow samples, where most of the low oxygen samples were coming from the deep water column. Same thing with density and salinity. And so what we're really seeing here is a signature of a deeper water column sample that ended up in the shallow part of the water column. And this was reflected as well in the microbial community. So just to wrap up this first part, we see that these water parcels do show distinct chemical and microbial signatures, and these signatures are driven largely by season and depth. We have some conclusions about the specific microbial taxa that we see, particular things like Nitrosopumilus, and a couple other clades that I didn't talk about so much and won't go into detail on, but we are very interested in why these particular taxa are showing up in these high levels in these times and places, and what that might tell us about the California current in different seasons and depths. So with that sort of foundation in mind, I want to go take this a step further and start to link our microbial and phytoplankton communities using shotgun metagenomics. And so, you know, as we think about the microbiomes of the water column, these are obviously very important ecologically and for, you know, for chemical and physical oceanographic reasons. But as we think about their impacts on higher trophic levels, we want to start to make those connections, and we're able to do that because we did marker gene sequencing from upper trophic levels as well as from the microbiomes, and so we have our 18S and CO1 marker gene data sets that tell us about phytoplankton. And so just to give a couple examples of why these connections are important, we have this sample phytoplankton group, diatoms. These are extremely important unicellular phytoplankton that are involved in a lot of primary productivity. In the California current, the typical paradigm is that during spring upwelling, diatoms are the first types of phytoplankton to bloom, fueled by this upwelled new nitrate coming from the deeper ocean. And they are your sort of case strategists. They can uptake this nitrate very quickly, bloom very quickly. And then as these blooms fade out over time, they are actually taken over by dinoflagellates, which is another major phytoplankton group. But these types of patterns and these upwelling patterns can vary a lot in different years and different seasons. Upwelling can be stronger or weaker depending on oceanographic conditions. And so this is certainly going to have an impact on our microbial communities as well. Just as another diatom microbe example, I mentioned this compound DMSP that bacteria can convert to DMS, which is volatile and will go up into the atmosphere. And there's been a lot of great work done looking at these sulfur metabolites that facilitate carbon flux driven by bacterial phytoplankton interactions. And so we're hoping to draw some connections from these by linking to these types of data sets together. And finally, I mentioned dinoflagellates earlier as sort of the next group of phytoplankton that take over after the diatoms bloom. And there's been some nice work done in Europe showing that diatoms and dinoflagellates, their sort of ratio to each other can be used as an ecological indicator of marine systems. And so these types of ideas are really driving the questions we're asking from these data. So just to recap our metabarcoding, these are our marker gene data sets that just give us community surveys. Contrast this with shotgun metagenome sequencing. This is when we sequence the entire pool of DNA, and then they go through a process called assembly, which tries to patch together short reads coming from the same longer piece of DNA back into longer contigs. And then finally, we try and bin these contigs into metagenome-assembled genomes, or MAGs. So we're trying to reconstruct entire microbial genomes, which can then link taxonomy and function purely by sequencing the entire eDNA pool. And just to go into this in a little more depth, we can actually learn a lot just from these short reads as well, just based on what's called Kamer composition. So this is just the ratios or the short sequences of A's, C's, T's, and G's that can be grouped together. And it turns out that these signatures are actually proxies for taxonomy. And so without doing any matching to databases, without doing any sort of complicated assembly, we can get a signature of the microbiome just by looking at Kamer composition. And I'm happy to talk more about that at the end if people are interested. I just wanted to briefly touch on it so you have a sense of what Kamers are. So as an example of this is now a similar ordination plot to the one I showed you before, but now it's based on these shotgun metagenomes, and the signatures are purely based on Kamers. So rather than matching sequences to taxonomic databases, we're just looking at the community signature of nucleotide composition. And it turns out this is actually a very good, very effective way to classify different samples and show their similarities and differences. And so here again, we see the same patterns. This is the same set of eDNA samples, just the metagenomes. And we see again that they're really separated by season and depth, as we saw before. So linking these to phytoplankton, how to go about doing this. So this is now involving multiple eDNA data sets, the metagenomes, as well as the phytoplankton communities as given by 18S and CO1 marker genes. And so what I did for these analyses was just to classify each sample as either high or low within each phytoplankton taxon. So as an example over here, if I'm looking at this particular phylum, the Bacillariophyta phylum, which is to which the diatoms belong, I said any samples that had above average levels of diatoms I classified as high, and below average were classified as low. And so this is kind of a crude and slightly arbitrary classification, but it's a starting point for where I wanted to go and see if any of these signatures can be linked to our microbial communities. So now I have these same plots, but they're now colored by high or low, whether or not the samples are high or low in diatoms on the left or dinoflagellates on the right. So if we focus on the plot on the left, we see that our spring shallow samples were all quite high in terms of diatom levels. The deeper samples, even from the spring, were low, and then in the fall, the shallow samples were also low when it came to diatom relative abundance. With dinoflagellates, the story is a little more complicated. We see essentially the inverse pattern where in the spring shallow samples were actually fairly low in dinoflagellate abundance, and the fall shallow samples were much higher. However, there is a little more variability here. So we do see that the fall shallow samples had some low dinoflagellate samples as well. But we start to get a sense of the differences in dinos and diatoms from these just by coloring based on high and low. So looking a little closer at the diatoms, I wanted to see if we could get to a little more resolution. So rather than just saying high or low, I classify them into 5 different groups. And again, we see that all of the highest groups, so 4 and 5s, are all in these fall shallow samples. But there is, again, a little more variability, and the relationship is not quite so clear-cut. So based on these patterns, can we actually use microbiomes to predict phytoplankton levels? So without knowing anything about the diatoms and dinoflagellates or anything about the chemical oceanography, can we just take a microbiome sample and use it to predict whether or not that sample will be high or low in diatoms and dinoflagellates? So to address this question, I use a machine learning technique called random forest, which uses decision trees to train a model set of your samples and then take that model and predict outcomes. And you don't have to worry too much about the features of these matrices, but essentially what I found was that the accuracy of the model for diatoms was fairly high, it's around 92%, looking at unknown samples. But for dinoflagellates, it was really only, it was not very accurate. So only about 2/3 of the time could it accurately predict whether or not dinoflagellates would be high or low based on the training sets that I gave it. So maybe the dinoflagellate story is a little more complicated, but nice to see that we can definitely see some correlations between diatoms and microbiomes. There were several other phytoplankton groups that showed some interesting patterns. I'm not going to focus on too many of them except for one called Haptophyta, which is over here on the bottom left. And what we see here is actually really interesting because we have some basic patterns where in the spring shallow samples at least seem to be fairly high. The spring deep samples are fairly low, but in the fall, we have a separation within the fall shallow samples. So even regardless of season and depth, there seems to be something else that is partitioning out these two levels of our haptophytes, and those are correlated with different microbiomes. And so this is interesting to me because, you know, we have these existing drivers that are season and depth, and we see that the diatoms fall out on those patterns, so do the microbiomes. Well, maybe that's a coincidence. The underlying factor we can't really know because they happen to be following the same patterns. But for this group, we actually see that within a season depth grouping, there is this separation, and that to me says that there's something distinct within that is separate from season or depth that is driving the difference in haptophyte level. And so when I looked at some random forest models for these groups, they were actually very highly accurate, with the exception of the arthropods, which was only 75%. But in particular, again, I want to focus on the haptophytes, where we were successfully able to predict haptophyte levels using only microbiome data. And this is interesting because this is really one family that was driving the haptophyte correlation, this family Neolerabdaceae. I apologize to any phytoplankton ecologists if I am mispronouncing that. But it includes this coccolithophore, Emiliania huxleyi, which is a really important phytoplankton. Coccolithophores generally are calcifying single-cell bloom-forming phytoplankton because they form these calcium carbonate shells, they do have implications for carbon cycling in the global ocean. And actually, conversely to what you might think, rather than drawing down carbon, they actually push the balance towards outgassing of carbon into the atmosphere. I don't quite understand the chemical oceanography there, but they certainly have important implications for carbon cycling. And in addition to that, they are, like many phytoplankton, big producers of DMSP. And so their abundances and their interactions in the global ocean are very important. Okay, so just to wrap up this second part, we see that these seasonal patterns in dinotom dinoflagellate levels can be linked to microbiomes. And these microbiomes can be used to predict the levels of certain phytoplankton groups, including coccolithophores, which are important members of the phytoplankton community. Okay, so in this third part, I want to really focus on the advantages of metagenomics, what we can see by sequencing shotgun metagenomes, going past just taxonomic composition and actually getting at gene function and linking that function to taxonomy using metagenome-assembled genomes. So again, for those of you less familiar with these techniques, I really want to drive home the point that binning into metagenome-assembled genomes is really, really powerful. This is a tree of life that was published a few years ago based on all the available genome sequences. We can see the bacteria, for one thing, really dominate this tree. And also that this phylum is incredibly diverse, and the vast majority of this tree has yet to be cultured or studied in depth. And so without the culturing information in a lab, we really rely on these culture-independent genome sequences to tell us who these bacteria are and what they're doing. So whole genomes provide much better taxonomic resolution than marker genes, which makes sense. The more DNA sequence data information you have, the more you can, the greater resolution you can get. And this is also because of horizontal gene transfer in bacteria, where things are not just evolving linearly, but they're actually exchanging genes. And the functional gene content that exists in these genome sequences provides information on biogeochemical functions. So in this example, we have our taxonomic marker gene, which tells us the taxonomy, and then we can link that in a whole genome to something that is actually performing a function in the ocean. In this case, I have an example of proteorhodopsin, which is a light-driven proton pump involved in converting energy, and a gene involved in this conversion of DMSP to DMS. This is just a cartoon example, but these are the sorts of data that we can get with MAGs. And finally, we can get more accurate abundance information. So rather than just looking at a single gene and its proportion in the community, we can actually take all of our short reads from the metagenome and map them back or align them back to our larger genome sequences. And we can see from their coverage levels how those abundances compare. So putting all of that information together, I know that's a lot. Oh, right. So sorry, just to mention some of the limitations. There are a lot of computational challenges associated with assembling and binning metagenomes. And this is a really active field of research that I've been working on for a while now. But it's a really exciting field, and we've made a ton of progress just in the last few years. And I think I mentioned earlier that some clades like SAR11 have very diverse populations, and this makes them very hard to bin into a consensus genome. And so this is really limiting the information that we can get from some rather important bacterial clades. And finally, some marker genes, including the 16S, rarely assemble well due to just repeats in their sequences, and that can be a limitation as well. Nevertheless, metagenome-assembled genomes have really changed the way we think about microbial communities, especially in the ocean. And when we bin and annotate these MAGs, I'm not going to go over a couple of these methods, but this is what gives us a more complete picture of who these microbes are and what they're doing at what levels. Okay, so with all of that information in mind, I'm going to show you a figure here that is very complicated, but I'll walk you through it. It's actually not as complicated as you might think. This is looking at 85 different metagenome-assembled genomes taken from 61 different samples from the California currents. And the center of this figure is not a phylogenetic tree. This is just a dendrogram where each branch is a sample or a metagenome. And each row going around the circle is a different mag or a different genome. And the level of the black bar in each row shows its relative abundance in the community. And then on this side over here, oh, where's my pointer? There we go. On this part of the plot up here, we've put in the annotation information for each genome. So we're identifying genes that were in the genome, and these genes can tell us a little bit about what these microbes are doing. And then finally, the outer layers of this circle plot are metadata associated with the sample. So in this case, I just have depth group and season, which are our two major driving forces in these metagenomes. And you can see right away, if you follow those color bars, there's a separation, a big separation between the shallow and the deep samples, as we saw in the ordination plots. And there's also a separation by season. And the separations are not the same. So we have, for example, this like intermediate little section that is very distinct from the other two sections, which is the shallow spring section of the plot. And finally, we have this outlier sample here where we see, and again, it's the same outlier sample that was a shallow water column sample with more of a deep water signature. And we see, again, it's kind of set apart from all of the shallow samples. And just as an example of how the functional information can inform these plots, I've highlighted here the gene for proteorhodopsin. This is a light-driven proton pump. Because it is light-driven, it really relies on sunlight, energy from sunlight. And so we see that the taxa, the mags that have this gene, are almost all absent from the deep water column samples. So in genomes where proteorhodopsin is present, those genomes are in very low or low abundant or absent from the deep water column samples, whereas they're very much more abundant in the shallow samples. And this makes total sense when we think about what taxa need to get energy from light. Okay, so this is just the way that we're thinking about these microbiomes and trying to look for some crucial biogeochemical functions that they're involved in different types of samples in the California current. So remember I showed you which are the top taxa in each group based on marker gene data. Now we look at these data from MAGs, and I'm not going to go into this too much, but I just wanted to say that the patterns and the actual taxa that are represented are very similar between these two types of data sets, which is very encouraging. We like to see that these two different types of approaches can give us very similar data. In particular, the ranges of relative abundances from the spring shallow samples we see are much greater due to the patterns that I talked about earlier. So in conclusion for this part, we see that our metagenomes can link taxonomy to function, which really starts to tell us what is happening at a very fine biogeochemical level in the California currents. And this is the type of information that we now want to take and start to link in a new way to phytoplankton and to higher organisms. Okay, so in this last part, I just want to give you sort of a preview of where this project is going and how we're expanding these analyses into a time series. So I've accumulated data from the last 4 years, coming from various different cruises. You don't have to worry about all the different cruise names. I just want to point out that 2017, we have a bunch of Northern California samples, largely from the Monterey Bay area. This is the data set that I have already discussed, the one from 2018, which was a very narrow geographic range. But thanks to a really nice collaboration we have with the National Marine Fisheries Service, we have been able to go out on these fisheries cruises for the last few years as well and collect water column samples from a much broader range of the California coast. And so, so for 2018, we are working on sequencing many more samples from covering the California current all the way up from Northern California down to Southern California. We also have a similar type of data set from 2019 and from 2021, the most recent year, where I was lucky enough to go and join on this field sampling and get a bunch of different samples covering this broad geographic range. And we're really hoping that this time series is going to really expand on the patterns that we've already seen just from this small 2018 data set. You know, we can start to really build out the time series and the geography and the season depth patterns that we see within this really productive and valuable marine ecosystem. And so as I mentioned, this past year I got to go participate in the RREAS cruise, and these are just some photos of me sampling water. So we use water from CTDs taken at various depths. I have my own little like eDNA processing lab on the cruise. And another great advantage of this cruise is that we can get trawl data from many of the sites. Sometimes we were not able to do trawls and I only got water column samples, but in many cases we have associated fisheries catch data from these samples where we're doing the sequencing, and that's going to be a really valuable component to this time series as well. So just a little bit of a sneak peek where we've combined 2018 and 2019 data. So now this is from 2 different years. And again, this is a metagenome k-mer-based distance plot. And what we can see again is that these separations by depth are very clear. There is even some separation by year, particularly in these intermediate water column depth samples. So our shallow and intermediate samples, particularly our most shallow samples between 0 and 50 meters are separating by year as well as by depth. And so the one limitation here is that 2019 samples were only taken in the spring, so we don't have the seasonal resolution for 2019. But we're starting to build again these larger time series, these larger data sets that can tell us about space and time patterns in the California current. And finally, I just want to mention that we do have chemical oceanographic data as well, and we can start to look at how microbiomes are partitioning out by nutrients, things like silicon, which is the one highlighted here. Nitrates, very important nutrient for phytoplankton and for microbes. Chlorophyll, of course, so we see where primary productivity can be really linked to different microbial communities. And finally, again, this is just a similar plot to one I showed you before, but for 2019. And so our goal is to really start to integrate all of these metagenomic genome samples, all this information, and partition our water column samples by microbiomes, which can then be linked to phytoplankton and hopefully ultimately to higher organisms, including fish. So as a final conclusion, we have these seasonal depth groupings that differentiate our water column samples in the California currents. We've seen that microbiomes are correlated with different phytoplankton, including diatoms and coccolithophores, some really crucial phytoplankton taxa driving processes like primary productivity. And finally, metagenomes let us link these genomes to taxonomy and link taxonomy and function within within a microbe and tell us, not only who these microbes are, but what they're doing and why it might be important for the rest of the ecosystem. So just to bring you back to our original study questions and goals, we are hoping to link these microbial communities not only to oceanographic processes like upwelling, but also to higher trophic levels, including plankton and metazoan communities, and use these data to ultimately inform predictive oceanographic models related to climate, related to fisheries. Microbes, as a microbial ecologist, I strongly believe they are the linchpin of these ecosystems, and if we learn what they are doing and when and why, that is really the key to understanding all the rest of the marine ecosystem. And so I think I'll end there. I'm definitely happy to take any questions. All my contact information is here. And I've also listed all my affiliations, which I didn't mention at the very beginning because they're a bit complicated. But
all right, thank you
there's
so
my
much,
Twitter handle as
Nastassia.
well.
Your research is super interesting. I did really appreciate your overview of metabarcoding at the beginning, not having any kind of omics background myself. So with that, it looks like we have some questions that are trickling into the chat. So to our audience members, feel free to start adding your questions here. Our first one is from Emma. Is any of your machine learning work published?
Thanks for that question, Emma. Unfortunately, none of this work is published yet. I've only been working on these data for really less than a year. And we're really hoping that the expanded time series is going to provide much more robust information. If you've worked with machine learning in the past, you know that the power is really in the amount of data you can feed it. And so thus far, it's been a fairly small data set, which means that the results that we see are really encouraging. The fact that we see these connections and these strong models, even with such a small data set,
All
to me
right,
says
and
that,
Emma, also we are working
with an
on
expanded data set, we're really going to have even more
NOAA
powerful
affiliated
conclusions. So
publications
thank you for
put
asking
up
that,
on
but
our
the answer is
NOAA
not
Omics
yet.
website that I dropped into the chat earlier. So stay tuned for that, and hopefully we'll be able to feature Nastassia's work then. Okay, it looks like we have a question that came in from Rao. Excellent presentation, Nastassia. Did you make AI predictions with comparison with the conventional methods or just genomic data?
I think you mean maybe metabarcoding versus metagenomes. And so right now, the machine learning data I showed was only from combining the 16S and 18S marker gene data sets, or in some cases 16S and CO1. But so that is just using like community composition based on amplicon data. Now, I could easily translate that into using relative abundance data from the metagenomes, and that is definitely on my to-do list. It's just quickest and easiest to use the metabarcoding data first, but we're hoping to
do we
take
have
that to the next
other
level with
questions
the metagenomic
from the
and mag
audience?
relative abundances
It looks like we have a couple
well.
people typing in the chat, so I'll just give folks here a minute. We do still have about 10 minutes or so, but could always end a little bit early if we're not getting questions. But I do want to give these folks a second to type their questions into the chat. Oh, okay, here we go. All right, so we have a question here from Tracy. Are you going to conduct this work in other large marine ecosystems?
I personally have my hands full with the California current right now. But at AOML, we are really interested in expanding our eDNA surveys to be global. And so we have some existing and upcoming collaborations with international and other larger survey efforts. So I'm actually going to be participating, for example, in a cruise run by the Tara microbiome, the Tara Foundation or group. If am you familiar with that? I'll be going down to Antarctica for that cruise and collecting eDNA samples there. And so that's just one example. We have other members of AOML participating in other cruises, oceanographic cruises all over the world, and we're constantly trying to incorporate eDNA sampling into these cruises. And so the ultimate goal, yes, is to build up more of a global database.
Thanks, Nastassia. We have a question that came in from Jeanette. Great presentation. It's great to highlight the many roles that microbes can play in the ocean environment. Totally agree. Could you further speculate on the possible impacts that they could have on fisheries management?
Sure, absolutely. Yeah, I've been trying to reach out to my fisheries colleagues and get a better sense of what are fish really? What do they need? What do they want? And the sense that I get from that community is that we can, by focusing on the lower parts of the trophic web, which is why I talk a lot about phytoplankton, we can think about the the prey that is available to particularly juvenile fish, adult fish as well, but largely the success of juvenile fish populations, depends on the nutrients that they can get from their food, which translates into the success of the population later on. And so, I'm trying to think a lot about how little phyto and zooplankton that are feeding these little baby fish swimming around, how those are being driven by microbial populations, and so in turn, if the phytoplankton community or zooplankton community shifts from one particular type of taxon that's dominating, let's say from coccolithoforested forested diatoms, for example, or from krill to copepods, or if those kinds of shifts would affect the juvenile fish prey field and what that would mean for the success of fish populations down the line. So those are the types of ideas that I've been thinking about, but I'm hoping to continue those conversations and
Awesome.
really
Thanks,
learn
Nastassia.
from my
We have a question that came in
metazoan
from Jeff.
focused colleagues.
Thanks so much,
I've
Nastassia,
just
for
been
a great
thinking about microbes
presentation. Sorry if I
exclusively for a
missed
long
it,
time.
but with all your sequencing efforts, does it seem like most bacteria species are represented in current databases, or are we missing a number? Seems like your data will help discover lots of new species. And I also realized I skipped a question from Tracy, so I'll go back to that after this question.
Yeah, thanks for that question, Jeff. It's whenever you do environmental sequencing of microbiomes, you always find new things. That's just the kind of the name of the game in this field. The diversity is so vast, and our knowledge is still quite limited. That said, there are a number of marine metagenomic data sets that are available in public databases, and they just haven't been really annotated or analyzed very well. And so a lot of sequences will be represented in like publicly available data that just exists as like deposited sequence data, but there's not really anything known about what those data represent. And so in terms of like entirely new to science taxa, the general principle is like I'm not discovering necessarily new sequences of DNA, but we are always constantly seeing things that are very poorly represented in databases or have very poor annotations. And so, yes, discovery is always it's always on the books. But I think it's a bit of a different line of inquiry to try and figure out brand new organisms versus trying to link things that we know something about to other things that we know something about, and try and draw conclusions from those data, if that makes sense. But it's always there in sort of the back of our minds, and certainly I'm constantly generating genome sequences that have no close relative, so that's just a testament to the vast diversity.
So it looks like Tracy just put something here in the chat, but I'll go ahead and read off her question anyway, and I also encourage other members of our audience to chime into the chat as well. Have you or anyone else looked at co-occurrence of these plankton communities with fish, seabird, and marine mammal guilds/groups?
Yeah, that's a great question, and we do have those data. So the 12S marker gene is typically what we would use to assess both teleost fish as well as some other higher organisms. And we do have those data, and I haven't really delved into it very deeply for a lot of the reasons that I talked about earlier. In my mind, there's just it's a very big leap to go from microbes to fish. There's just a lot going on in the middle there, in between. And so the first things I wanted to focus on was looking just sort of one or two steps up from microbes and see if we can see patterns there. I do think a natural next step, a very important next step, will be linking things like phytoplankton and fish. And there has been some work done on that already, not by me, by others.
Thank you so much,
but this
Nastassia.
is definitely
we have
an
somebody typing in the chat, so just give it a moment to see if we have other folks who'd like to ask questions. Oh, here we go. Okay, we have a question from Patricia. What kind of sequencer are you using? Are there commercial kits available, or are these something that you are developing?
Yeah, so we largely use Illumina sequencing for both the marker gene metabarcoding sequencing as well as the metagenomics. And uh, we typically send out our samples to either university core facilities or other big sequencing centers. There are labs that have their own sequencing instruments. It's not like I know the Southwest Fisheries has I think a MySeq that they run in-house, and so you can definitely do that. It does involve quite a bit of time and effort to run and to maintain a sequencer, and also the technology is always changing, and so you kind of have to stay on top of that. And so we send out our samples to sequencing cores, but there's this very like commonly available kits both for library preparation and for sequencing. You know, Illumina has really cornered the market, I would say, in terms of these
All
short
right,
read
great. All right, we have just like a couple minutes left. Are there any other members of our audience who'd like to ask questions? I'll just wait a couple of seconds to see. Okay, so I don't see anybody typing in the chat, so I think I'm going to go ahead and wrap up our seminar here today. So I want to thank you all so much again for joining us today, and thank you, Nastassia, again for presenting for the Omics Seminar Series. Just a reminder to our audience, the NOAA Omics Seminar Series takes place on the third Wednesday of every month at 12 p.m. Eastern Time. Our next seminar will take place on December 15th, 2021. I think I dropped this in the chat already, but I will do so again. Here is the link to the NOAA Omics website, which has information about our next seminar, including the registration. Our next seminar is titled Environmental DNA and RNA for Advanced Management and Accurate Surveillance of Aquatic Invasive Species. So Dr. Subha Rao Chaganti from the Cooperative Institute for Great Lakes Research will be presenting on that day. So like I said, registration is now up on the website. Thank you all again for joining, and have a great rest of your day.
Thank you.